This material is made available to you by or on behalf of the University of Melbourne under Section 113P of the Copyright Act 1968. It may be subject to copyright. For more information, visit the University Copyright website.
That was, I was, not that I'm surprised, that level of detail is in the textbook, but it was such a nice summary.
Yeah, exactly.
Yeah.
It's different from something I've seen before as well.
Because usually I do try and find a publication or something that shows an example, but when it's in the textbook...
Why not just take it, it's that right there in front of you, why not just take it?
Yep. So today's lecture probably will be a little bit short, and you may have heard of a lot of this information in other subjects, but it was more also to reiterate that you need to know it in the context of biomaterials, and to, I guess, put it also into context with biomaterials and medical devices. So ethical issues, so not just ethics, but ethical issues with medical devices and biomaterials. So really, the ethical principle in biomaterial research is the protection of patients. We're developing things to either make their life better, make their life longer. You're not developing things to harm them. And then when we're developing the materials, we're wanting to do it for the protection of patients. So how do we ensure that? There's going to be standards, there's going to be rules or guidelines to follow. So this, how it's written, is very much FDA style, so very much the Food and Drug Administration in the US. There are some slight wording differences for the TGA and different regulatory bodies around the world, but the three, I guess, types of guidelines or principles are roughly the same. They just may come under different headings whereabouts you are. So scientific investigation into the development of medical devices should. So the preclinical studies must use good laboratory practice. The manufacture of devices must follow good manufacturing practice. So the wording is slightly different for Australia, and I'll go into details in the next few slides. And then clinical trials must be in compliance with good clinical practice. So, has anybody worked in a lab where they have GLP or GMP facilities? So I did my internship in a pharmaceutical company in their R&D department. So where I was, they didn't have GMP, but they had GLP, and then you got to go see the GMP facilities. You have to dress up, well, not dress up, but put on you don't just wear a lab coat, you have full-on gowns, everything is covered, your shoes are covered. You don't often see those sorts of labs in universities, they're more out in industry. But if you're wanting to develop something for a medical application that's going to go into patients, you're going to need good manufacturing practices. So what does good laboratory practice mean? It's essentially a quality system. So like with a lot of the things that you would have gone through in clinical trials and regulations, a lot of it is about paperwork and keeping track of things. But for good laboratory practice, it also applies to certain things. So personnel involved, you usually have a chief investigator. It'll outline what their role is, the people who work on it, what their role is and what they need to do. The equipment and facilities that you have in there, quite often there'll be protocols for how they're maintained, how they're calibrated, how you run the experiments on them, protocols for running the experiments that you need to carry out, and record keeping. So a lot of it is about having a system in place so it's documented. So when you go to the TGA or FDA or whatever regulatory body and say, I have done all these studies to register my device, or I want to go put it into a clinical trial, you have evidence that you have done it correctly. So with regards to good manufacturing practice, if you have a look at the TGA website, and you should have gone through this last semester with regulations, so you have good manufacturing practices for medicine and good manufacturing practices for human blood tissues. But for medical devices, it actually comes under the conformity assessment. I tried to read a lot of the documents over the past few days while I was putting the lecture together and just having a look at the background and reading what the FDA and other regulatory bodies have. A lot of it is the same wording. There are going to be slight differences, but to know in Australia, your medical device has to pass through the conformity assessment and you have to follow that set of guidelines rather than necessarily, it's a little bit more about wording of what it is called, but you still have to do the same sort of things. And it's the same sort of things with GLP. It's a lot about record keeping, personnel qualifications, so are they qualified to be carrying out the sort of assessments that you need to do? Sanitation, cleanliness, so that's, yeah, how well you keep your equipment, people coming into the facilities as well. I used to find, I guess if you worked in it every day, you got used to it, but walking past the first stage into a GLP lab, you go over a sticky bit, so anything that's on your shoes will come off, and you pretty much get suited up, and then when you go into the lab, it's, yeah, the regulations or I guess how they've got everything set up, it's, sorry, what am I trying to say? The paperwork and the procedures that you need to go through in a GMP facility is very, very tightly controlled, very, very regulated, and you need to make sure that all those records are kept so that, again, you have proof that this is how everything has been done. So I put, having a look at the Good Clinical Practice Australian website, this is the first thing that you'll see when you go onto the website. So the principles of good clinical practice have their origin in the World Medical Association's Declaration of Helsinki. And that's just essentially saying, well, they brought that into place to ensure that all human studies that were carried out so that the participants have their own rights or still have rights. Safety and well-being is placed above all other considerations involved. So any regulatory body or any body that you go through to get approval to do particularly human studies, what you say you're going to do in your studies has to follow the Declaration of Helsinki, and that's how the questions that you answer in the ethics application and what you say you're going to do, they will make sure that it complies with that. So the TGA has essentially adopted a European Union version of these guidelines in Australia. And then I've put, there's some standards as well with regards to the design, the conduct and recording of your clinical trials. I'm not going to go through how to design a clinical trial because I'm not saying it's not associated with biomaterials and medical devices, but that's not the main point of this course. Just knowing that when designing medical devices, these are the sorts of things that you're going to need to go through. So I've put this slide up here to think about the two different sides of people who do clinical trials. So you have your engineers, and sometimes, well, not sometimes, they can be scientists as well, who are developing the technology, innovating the different ideas about the devices or what we're developing to help a patient. Whenever I have done any work that is going to be translatable, it has always involved a clinician. Because you've probably heard before, if you're developing these technologies, if you haven't talked to the person who's going to use it, if they don't say it's worthwhile, it's pretty much useless. So if you're having a clinical trial, or you're doing human testing or need to put in human ethics, the likelihood of you having a physician involved that's going to also be an investigator is very high. But then if you think of the different things that they think about compared to us, as an engineer or a scientist, I like to see statistics. I like to see that something has changed, and if it has changed, it's significantly different to something else. And I'm the sort of person where I go into the lab before I do experiments. I have my protocol ready, and I follow it, and then I do my experiment, and then I know if I'm going to repeat it again, I follow the same protocol so I can repeat it, and I can show that just because I've done it one way, the results that I get the first time, if I do it a second time, it's going to be the exact same way. So I'm not saying that physicians don't do it that way, but sometimes they have a different way of thinking. They're always going to be guided by what they see in their clinic, their expertise, their everyday occurrences. But the most important thing is that they're always going to do stuff to ensure that it's providing the best outcome for their patient. So if you're developing something, and as fancy or as technologically advanced it may be, if it's not going to provide the patient with something better than what they already have, then they may not support what you want to do. I guess it's always things to think about when you're developing things, when you're talking to clinicians about what both sides of the party want. And this, because with clinical trials, how you write the clinical trial of what you're going to do and the outcomes, then that's, I guess, I'm not saying as engineers or people that have developed it, we don't want to see the patients improving, but we also want to test how our device is going. Whereas with a clinician, at the end of the day, the patient safety is the first thing on their mind. So whether it's in sometimes preclinical, your in vitro, your in vivo studies, before you even go into a clinical trial, you may need to put in human ethics or animal ethics to do some testing. So I've given, in the next 2 slides, I'm going to show you some examples and talk through a few things about what you need to put in an ethics application. I'm not going to show you an example. Ethics applications are hundreds of pages long. There's a lot of paperwork that you have to go through, but I've noted some of the main points that you need to tell them when you're putting in the application. So for a human ethics approval, it's required for all levels of research you conduct. So even research, if you are carrying out a survey on someone, if you're doing it for research, you have to put in an ethics application because essentially you are collecting someone's data and you're going to store it and you're going to do something with that data. So whether it's a survey or whether it's collecting a sample from patients or you're getting humans to do it, an ethics application has to be put in. So what I think are the main elements of a human ethics application. So you have to state up front what your aims and your objectives of the study are. You need to tell the ethics committee why you want to do this. And it can't just be because I want to. There has to be an objective reason for doing it. You usually have to outline whether or not you want a specific population. So whether it's got to do with ages, so kids, teenagers, adults, elderly, depending on what the study is, that may need to be taken into account. If you're doing a study on a particular device, does the age of the patient matter? Ethnicity, sometimes that can matter. So I know with medical devices, particularly if you're trying to get a medical device into China, they're starting to change their regulations. Before, if you wanted to have a medical device in China, even if you had gone through all the regulations in a country outside of China, you still had to do the same sort of studies inside China. They're now saying if you've done studies that comply with what the FDA asks, as long as your trials that you have done contain a population that's equivalent, or you've done studies on, I guess, Chinese ethnicity that can demonstrate that you've tested your device on a population of Chinese, so that then they could say that you don't need to do the studies in China as well. So that's an example where ethnicity may come into account. There could be other examples as well. So, access to databanks. So for some of my research, I look at different disease states. So rather than going to, particularly in a lot of the hospitals and around Victoria, some other places in Australia, obviously the world, there are a lot of data banks for tissues. So patients that have gone in for whatever reason, a biopsy may have been taken, there are a lot of tissue data banks available. So if I wanted to compare a whole range of different, so here's an example. I develop materials for wound healing. So people with diabetes have different types, have slower wound healing than people without diabetes. So I've gone and taken skin samples from diabetic patients and compared those cells to normal cells. So that's where a tissue bank, I would still have to put in ethics applications to get those cells, but that's an example where a tissue bank would come into account. So of course you have to detail the design of the experiments, particularly with patients. How are you going to recruit them? Do they get anything for their time? Sometimes they do, particularly a lot of the time for surveys, the incentive is, if you do this survey, you get a so-and-so voucher, or if you do this survey, you go into a draw to win. What the participants have to do? What is going to happen with their data or the material that's collected? If you're taking a biological sample, once you've used it, do you dispose of it? Do you store it? How long do you store it for? And then how you're going to analyse their data and where their data may be available. You then have to put your risks to benefits and how you're going to monitor them, and then how you're going to obtain consent. So have a think about that one. The one difference between animal ethics and human ethics is humans can give consent. They can say yes or no as to whether or not. Which is why sometimes it's a lot easier to get human ethics than it is to get to animal ethics, because you have to show, and I'll go through the animal ethics, how you monitor the animals to make sure that what you're doing is okay. So with regards to animal studies, and if you're putting in animal ethics, the two basic ethical principles of animal ethics, relative to the use of animals in biomaterials research. So firstly, the experimentation on living animals should be reduced to the minimum necessary to obtain the necessary scientific information. And secondly, the pain, distress and other harm to the animals should be reduced to the minimum necessary to obtain valid scientific data. So I guess what do those two principles mean? When you put in an animal's ethics application, if you're doing an implant study on a sheep with hip implants, you can't say, I want to do 50 sheep. You need to say, you need to do You need to show your experimental design to how you justify the amount of number of animals you need to carry out the experiments so that you still get valid scientific data and valid scientific information, but you've shown that you've made sure you need the minimum really required. So you're not using too many, but you're using enough so that you're justifying why you need that number of animals. So when I talked about some of the in vivo studies, one of the, I forget which standard it was, but one of them said if you're doing a long-term study in sheep, you should have this many animals, this many female, this many male. If you're doing a long-term study in mice, you should have this many animals, this many female, this many male. So it's using that sort of scientific, that information from standards to then help calculate how many animals you need. So when you put in an animals ethics application, you obviously put in what the project is, what the aim of it is, what you're going to do in the experiment, the number of animals, the type of animals. You also then have to include how you're going to treat the animals, how the animal is going to get to the facility, how you're going to house them, how are they going to be acclimatized beforehand as well. So there's a whole lot of information that you need to include about how the animals are going to be treated both before and during the experiment. Particularly how you're going to be monitored, how they're going to be monitored, so before I did animal experiments, I had to go do an animal ethics course, and they teach you how to monitor if an animal is distressed, particularly while you're doing surgery, they're going to be under anaesthetic. How do you know how to monitor an animal? It's similar in a way to a human, but it's going to be different. So you look for vital signs, things like that, but you need to be able to show that in the ethics application. And then the fate of the animals. So what's going to happen to the animals after the study has finished? You need to, in the application, justify why animals are required for the work. So that's where, if you've done the right preclinical in vitro studies to show that this material is safe, it doesn't do this, it doesn't do that, it does what we want it to do, we need to now go into an animal study. It's those sorts of preclinical evidence to show why you need to move to the next, why you need to move into animals before going into humans. Justification for the choice of animal. So again, if it's a particular device and they usually use a particular animal breed because of its physiology, its anatomy, that's relevant to that device or that material, that could justify. And then you need to show how many animals you require. So actual evidence, calculations, you're going to do this many animals in this many repeats to really show that you have designed a study so that you're minimising the number of animals required, but still able to get scientific data out of it. So really, this is just reiterating a lot of the regulatory bodies that are involved with this. You've heard of the FDA, you've heard of the TGA. So a lot of the standards that I show you, quite often you'll see an ISO number in front of it. So ISO stands for the International Standards Organization, but there are a few other ones. So there's the American Standard for Testing of Materials, Advancement for Medical Instrumentation, and American National Standards Institute. A lot of the three of them will get their information from the ISO standards, or they are quite the same. But it's more, if you're seeing these acronyms, and you probably would have seen a lot of them, if not in these lectures, maybe in the ones before I started lecturing in the subject. But these are the regulatory bodies, or they govern the standards that we use. So this is the last thing that I wanted to cover. Do you guys have any classes or any subjects where you go through intellectual property and that sort of thing? Yes, no? Not yet. You might. You might do it in management. You probably, if you choose, is it by innovations design subject? If you choose that, you probably will find out a lot more about it. So, because I work in academia, I often hear the publish or perish, patent or perish. So for me, for most of my research, my main objective is to put it into the scientific literature. Whereas with a company, their main objective is usually to patent everything. So then what's the difference? So a patent is really you're protecting your intellectual property. So it's the protection of one's ideas that have potential for earning profit. So it's not my complete area of expertise, so I'll go through a little bit of this that I know. But it's really you're protecting your idea. So while I say in academia it's usually to publish, it's not always. We can patent things, particularly if the research that we do is with a company. Their main goal may be to patent it, particularly if we want to make something, if we want to commercialise it. So a lot of the time that comes down to record keeping, to show what date you came up with the idea. But a patent is really your exclusive rights to the idea. You can then license the patent to someone else and they give you money, but you usually have it for a set period of time. So a lot of the time you see, or what I really associate with things coming off patents, you quite often see it in the pharmaceutical industry. You'll have a particular pharmaceutical that is sold by one of the big companies, then 10, 15 years after it's been on the market, all of a sudden you see all the generics come out. So that means that that particular patent on that pharmaceutical has stopped. Anybody who wants to make it can make it, because the other thing with patenting is that it then becomes public knowledge. You have a patent which you can then, it goes online and you can search people's patents. So you can see what intellectual property they have. You just, if you want to use it, have to pay them for it. And if they find out that you're using it without that, they can take you to court and you can get charged with infringement. So it's a way that companies are able to really put, to a degree, monetary value on their intellectual property or their ideas so that they're able to generate a profit out of it. So one example of this is, have you guys heard of CRISPR? So the gene editing tool. So there must have been something with regards to who developed CRISPR first. And I only know this because I want to do CRISPR stuff in my research, but to do that, the University of Melbourne, I can only go with one company because there is a dispute between two different universities that this came out of to who actually owns the technology. So who is able to, I don't know the exact lawsuit, but I'm thinking that someone hasn't patented it or they're in the process of patenting it, and because this technology has taken off, they can actually make a fair bit of money out of it. So it's one of those things where record keeping comes into a big play. If you have two companies that have come up with the same idea around the same time, how do you show which one came up with it first? You go to their lab books, or you go to where they write down their ideas and their data or the experiments of how they've come up with this, and you see who did it first. So that's where keeping record of these sorts of things is crucial from any sort of stage. So whether or not it's you're doing the tests or for preclinical studies, or you're wanting to patent something, record keeping is a very big thing. So that was all I have for this lecture. I said it was going to be a little bit short. Does anybody have any questions?
Applies to an update applies to the US that changed a whole bunch of laws about copyrights and patents recently, like extending them and stuff. The whole point, the Disney case, if you know what I'm referring.
Probably.
That potentially affect our industry.
I would have to have a look. And the one thing about regulations is it changes so frequently. And that's, I was talking to someone from the Chinese FDA, and they were even saying how when companies in China are putting in, I want to put in a medical device, the paperwork that they've started doing, all the tests from 5 years ago, because that's how long it takes, things have changed so fast that by the time they get to, I want to register this medical device, things have changed so much that they're not doing it the right way and they have to go back and do it again. So yeah, it's one of those, it changes so quickly, but can make such a big impact on what you're trying to do. Okay, the next few lessons after this should be a little bit more exciting because we'll look into different applications where biomaterials are used.